This video is meant to introduce DNA and its structure. We're going to start by looking at Griffith's experiment. Now, he took a bacteria, we're just going to call it S bacteria, and he injected some mice with that bacteria. They all died. Well, later, he decided, what if I heat that bacteria and kill it, and then inject them with the dead bacteria? And so he did, and the mice lived. He then chose a different bacteria, R bacteria, and he injected the mice with the R bacteria, and the mice were fine. But what do you think would happen if he injects them with the live R bacteria? So they were okay the last time he injected them with the live R bacteria, and with the dead S bacteria. Last time he did that, they were okay. He combined those two, and now he injects these mice. This time, though, he finds that they're all dead. So he concluded that there was something in the dead S bacteria that could affect the live R bacteria. But what was it? Was it a protein? Was it DNA? What was it? Well, later, Hershey and Chase did an experiment. In their experiment, they wanted to test what carries the genetic material, DNA or protein. So they got a virus, and they coated the protein part of the virus with a radioactive isotope. And then they coated the DNA of the virus with a radioactive isotope. And they wanted to see what would happen. So in the case of the protein coat, they found that there was no radioactivity inside the bacteria. But the DNA, they did find inside the bacteria. So they concluded that it must be the DNA that has this genetic influence that can change the traits of something. So that raises some questions like, what is DNA? Well, one way to kind of think of DNA is a chemical molecule, but it's like a book. How's it like a book? Well, it's like a book because it stores genetic information. It's like a book because it can be copied. And it's like a book because it transmits that information to other places. So for humans, we have 46 chromosomes, 23 pairs. That's our genetic code. So that's like our entire book. All right, our genome, the whole thing. But the individual chromosomes are kind of like chapters. So we would have 46 chapters of 23 pairs. And then genes code for individual traits. And those would be like individual recipes in our little cookbook example. DNA, RNA, ATP, they're made of nucleotides. Nucleotides are the monomer for the DNA, which is the polymer, or RNA, which would be the polymer. The nucleotide would be the monomer. It consists of three basic parts. A is a phosphate, B is a sugar, C is a nitrogen base. Those are the three major parts of a nucleotide. That's our monomer, our individual piece. Now those nitrogen bases, we could have one of four nitrogen bases: adenine, thymine, cytosine, guanine, A, T, C, and G. The question now is that we've identified DNA as a information-bearing molecule, and it has these nitrogen bases. Chargaff was a scientist who was trying to figure out, how do those bases convey information? And he found out that the bases pair. But how do they pair? So if you're looking at this hodgepodge mess here, we know that these bases pair, but how? So count the number of T's, for example. So 1, 2, 3, 4, 5, 6, 7, 8 T's, 8 thymines. Well, if there's a pair, that means there's 8 of something else. So what else is there a pair of? What else is there 8 of? In this case, there's 8 adenines, A. So A's and T's pair, C's and G's pair. And that's kind of how he figured it out. We can remember this pairing by thinking of the straight letter, or the straight lines using the letter A, and the straight lines using the letter T. A pairs with T, they both have straight lines. C pairs with G, they both have curved lines. And again, we can that can help us remember. The A's and the G's are called purines. You'll notice that there's like the two rings in that molecule that are connected together. The pyrimidines only have the one ring. But purines pair with pyrimidines. G's and C's pair, A's and T's pair. So the way you can remember purines is this little mnemonic: all gold is pure. All adenine, gold, guanine, pure, purines. But why do they pair? Like, why do C's and G's pair? It's not because they have curved lines in their name, but that's how we can remember. But why do they pair? Well, they pair because of hydrogen bonds. C's and G's pair because they make 3 hydrogen bonds. A's and T's pair because they make 2 hydrogen bonds. So in between the 2 strands of DNA are hydrogen bonds between the nitrogen bases. But along the backbone between the sugars and the phosphates, those are covalent bonds. So they're much stronger bonds. The red dots there are the phosphates, and the blue pentagons are the sugars. So this is a Platt-Shaw's rule. If what he did, he worked with percentages rather than individual molecules. But if a species has 35% adenine, what's the percentage of the other bases? Well, we know that A equals T. So if adenine is 35%, then thymine is 35%. We add them together, we'll get 70%. Now, if we have 70%, we need up to 100%. So we find out what's left. So 100% minus 70 is 30%. So that 30% is going to be split between the C's and G's. So we're going to just take half of 30 for each C and half for G. And that's going to give us 15 for the others. So those experiments were to help us figure out what's in DNA. Now, the next major thing to discover was the shape of DNA. And this was something that Rosalind Franklin, Watson, and Crick worked on to try to discover. What they discovered was it was a twisted ladder shape, this double helix shape. So just to review a little bit, this is a nucleotide. The orange ball is a phosphate. The green is a nitrogen base. The blue is a sugar. So now we know the shape, that is a double helix, this twisted ladder shape, how it stores information. It stores information by that, there's A's and T's and C's and G's. We need to ask these questions like, how is it copied and transmitted, and what are those things? Well, the information is stored in these unique combinations of A's, T's, and C's and G's. So not everyone has the same amount of A or the same amount of T. We know that those two are going to equal, but whether you have 10% A or 3% A, well, that's unique to each living thing. Also, the amount of DNA, like humans have 46 chromosomes, other living things have different numbers of chromosomes. It's copied because whenever cells divide, each cell needs its own set of DNA. And that DNA needs to be identical to the cell that it's copied from. Like skin cells, your skin cells are copying and dividing all the time. Each cell needs to be identical to the previous one. It's transmitted because DNA stays in the nucleus. It doesn't leave the nucleus. But that information has to get out of the nucleus to do its job, making proteins. It does that with this other molecule, RNA. So RNA is going to take that DNA out of that message from the DNA out of the nucleus so that proteins can be made. This is just about the structure of DNA and some of its functions. In another video, we'll look at how DNA is copied in that process of replication.